Okay, good morning. We will start the session today. I'm presenting Martin Weigt, sort of. Thank you.
So yeah, it's switched on. Good morning to everybody, and can you make it a bit like this? Oh yeah, like this is nice. Okay, thank you. In the beginning, I would like to apologize that I just arrived a day later, but at the end, it's almost by chance that I even arrived, because when I came to Buenos Aires, I switched on my telephone again, and I saw something like 5 messages by the university director, by the director of my lab, by the French Research Council, and so on. Don't go abroad. So I was just in the new immigration lane in Buenos Aires, so it was something like 8 hours too late. So now I'm here. Oh, sorry, this reminds me that I should switch off the cell phone, because if not, I will get 10 other messages come back. Okay, so sorry, I'm going to talk about protein interaction and inference of protein-protein interaction at multiple scales, and it's something which we spend quite some energy in my group, and that's why it's a bit a patchwork talk. It's a bit an overview talk over several approaches, and we are still fighting to get this into one big picture, but we are not yet there, as you will see. So the starting point is this picture, which is a picture obviously not by us, but by Peter Oetz's lab, who is one of the best-known experts in the high-throughput generation of these networks using yeast-2 hybrid experiments. And they have some years ago published this picture, which is still very well in its main information, maybe if some details may have changed. So here, this is a protein-protein interaction network of Escherichia coli, so a bacterial protein-protein interaction network, and here you have this typical hairball picture with a lot of proteins. Proteins are dots, and a lot of links, which are protein-protein interactions. But they have nicely colored this picture by putting, for instance, the idea if there is some structural information about the individual proteins. And you see in green all those which have a resolved experimental structure, in yellow those which have some modeled experimental structure, and so on. And there is also the gray dots which have no structural data at all. So you don't know the protein structure for those one, not even by modeling. And they have chosen some kind of optimistic coloring, that they have somehow hidden the one without structural data in gray, which you don't see so well as the others. But if you would just invert the colors and put them in red and say this is what where you still should do something, it would be also very red. Okay, so this is the one. But even if you know quite a lot of information, they have enlarged some complexes, and complexes in principle should all be come together. And you see that in terms of structural information, sometimes they have 2 together, sometimes a bit more together, but never all. And then there is another nice aspect, is they have put links for protein-protein interactions. In some cases, as this one here, you have the structural knowledge about this, but they have colored them also in 3 colors. By in yellow, those ones which are known in the literature, but which they did not find in their experiment. Their experiment is a yeast-2 hybrid experiment, was at this point the best experiment existing for E. coli. They put the ones they found in green, which were unknown so far, and then they put in blue the ones which were already known, and they found back. And you see it's a mess. You see it's a total mess. A lot is probably missing in the literature. They find new stuff, but maybe it's wrong, and so on. So there will be a lot of false positives, a lot of false negatives, and so on. So this the status still of finding these genome scale I'm not French, but the French flag is because I'm here, I'm German, but so it's even hitting me. Okay, can I put it away? Okay, so there is a lot, so from the experimental point of view, it's a system, there's still a lot has to be done, and this is even for a simple bacterium. It's much worse if you go to higher organisms. But I don't want to go to talk about higher organisms, but mostly about E. coli and bacterial interactions. There might be some alternative, and that's why I'm interested in this work, to do actually something with the flood of new data, in particular of sequence data. This will be totally about sequence data, which is arriving in biology thanks to modern sequencing techniques. And what I've shown here is just for the so-called UniProt database, which is the probably most well-known and most complete database of protein sequences. I've shown the total size of the database, which is this red line. And note, this is a logarithmic scale, so there's roughly an exponential growth with some particularities about the curation of the database, which the curators tried to make more sustainable for future, but it's growing exponentially, basically. And as in the example which I just showed you, there is some, also in this database, there is some other sub-database, the so-called SwissPros database, which contains all those proteins where you have some manual annotations. So these would be, for instance, the colored proteins before, because you know the structure, the red ones. Or the ones where you have some protein-protein interaction, or some functional annotation. So somebody has done experiments and has found out something on these proteins, and this was put into the database. And you see this one is flat, and it's really flat. It went something like from 500,000 in 2010 to 560,000 now. The knowledge gets better, but biologists tend to work always on the same proteins, which are proteins from so-called model organisms, and so we accumulate more and more information about a small part, but obviously the E. coli I showed you before is a model organism. Even there it's incomplete, but we also accumulate this enormous amount of sequences which we have just sequenced. So it's a sequence over 20 letters, which are the amino acids. The situation in principle is not so bad as I describe it, because you can organize the many proteins in so-called protein families. Typically this is not really done at the protein level, but at the level of protein domains, because domains have some frequently some modular architecture using domains, and the domains are organized into domain families. And a family is in principle defined by some common evolutionary origin, so some common ancestry. So these are proteins which are so-called homologs, and they typically have a very conserved biological structure, function, or what we are interested in, interactions, in the sense that these are frequently kind of doing the same job in different species, or sometimes the same job in different pathways in the same species. Nevertheless, in the course of evolution, they have accumulated a lot of mutations. Sequences are very diverged. So typically, if you take a bacterial protein family, you find something that to randomly chosen proteins have something like 20–30% of their amino acids in common, and all the others have been exchanged in the course of evolution. So there's a huge variability. These two proteins are an overlay of two proteins at 30% sequence identity, and you see structurally they are almost the same up to little rearrangements, but if not, they are almost the same. On the other hand, if you take one of these proteins and you do something like 5–10 mutations, random mutations, you can be almost sure that this protein will not work anymore. So you can introduce a lot of variability, but on the other hand, it's also kind of fragile. If you use random variability, you destroy it. So the variability has to be constrained. If the variability is constrained, we can in principle use some statistical methods to actually get these constraints out of the data and learn something about the proteins. So that's our program, basically. And what is nice, that there are some people, for instance, those curating the PFAM database, which provide us these protein families in form of multiple sequence alignments. And they look somehow like this. Here I have it done already for two families. So this is a part of one family protein multiple sequence alignment. It's some small ribosomal subunit protein. Basically each line here is one protein sequences, is one amino acid sequences aligned in a way that basically, if I go back here, something position like the blue and the orange position here should be in the same column. So each line is a protein, each column is a specific position, which in the best case would go back to the ancestor position in evolution. Okay, now my question is, if I take two of these tables, two of these multiple sequence alignment, what can I say about the proteins? Just staring at these tables. In principle, not knowing anything about biology in this case, so I'm doing something which is totally ignorant, but I want to just from the statistics of this table get information about these. Obviously, I cannot be totally ignorant because I have to have some means to stare at these tables, and I want to ask 3 questions. So here, this picture I will use a lot to symbolize multiple sequence alignments, and so each line here would be one protein, and if you look to these alignments, then you have inside one species frequently more than one of these proteins. So this is the case when you have the homologs in different pathways, so-called paralogs, and you have it in different species. Typically, this is more of the order of 1,000 lines in principle, but obviously this is some sketches. So now you could ask 3 questions. First one is there an interaction in general between the 2 protein families? What does it mean? It does mean that there are some proteins in the family 1 and some proteins in the family 2 which interact, and there's some evolutionary conservation of this interaction. Whatever this means in detail. But even if you know that these proteins interact, you still don't know who is directly interacting with whom. Protein-protein interactions are very specific in many cases, so it may be that the first one here interacts with the third one, but not with the 2 other ones, which are even in the same species. So can we get something about the specificity of interaction? And then to go even to a more finer scale of resolution, I showed you these complexes, the pictures of the structure. And in this structure, it's important to know what is in which residues are in the interface, which residues get into contact between the 2. So can we say that this residue here is in contact with this residue, just staring at these tables? Always just staring at these tables. We stare with some guidance, some simple biological insight about evolution and about co-evolution of proteins, which means that basically interacting proteins don't evolve independently because they have a conserved interaction, or if they have a conserved interaction. And this can be seen at several scales. The highest scale is something which is related to what is called phylogenetic profiling, which is a method which was introduced something like 21 years ago by Pellegrini et al. And the idea was, if you have 2 proteins which have some joint functionality, and you need both for them of the functionality, you either have both of them in the genome present or none of them, because one of them doesn't make sense. So you have some correlated profiles of present and absence of interacting proteins. So this is just, is a protein present or not? Is there a homolog? Is a family present or not in a genome? This is symbolized here. But then you can go to this finer scale of interaction specificity, which I told you is that if the red one is several times present, the blue one is several times present, which one is actually interacting with which one? This red one, for instance, with this blue one, but not with this blue one, and so on. Part of this is encoded in a genomic location and neighborship, which I won't use here, part not. And then you can go to this last scale, what I said to the residue scale is actually that you have some co-evolution due to the fact that if you have two residues which are in contact, it's really a physical contact. So they have to have some compatible physical-chemical properties. You cannot put any amino acid easily close to any other. For instance, you have charged amino acids, and it's not so easy to put 2 equal charges close to each other. Okay. So just to come back to remind you, this is exactly, in terms of co-evolution, I have exactly rephrased these questions.